Hello, my name is Gavin Duke, and here's my project on the development of an ER stress reporter zebrafish line. And so the goal of this was to utilize the QFQS promoter system in order to look at the XBP1 mRNA, which is only translated in cells that are currently undergoing ER stress. And so this will act as a direct readout of ER stress by tying GFP to that XBP1 mRNA. And so, in order to test the validity of this construct, we used heat shock at 40 degrees Celsius on larvae at 5 dpf. And basically, we just looked if there was an increase of green fluorescence compared to the controls, which were just non-transgenic fish. And so, the background of this is that ER stress is just when there's abundance of misfolded proteins within the ER lumen. This triggers unfolded protein responses within those cells, and this has been shown to lead to apoptosis and has also been seen in the pathophysiology of both types of diabetes, mellitus. And so it's interesting in order to look at how different genetic backgrounds and possibly different physiological conditions may lead to different levels of ER stress within humans, but specifically ER stress within zebrafish, which would act as a model for different genetic backgrounds as humans. And it would be very interesting to see. So that was the goal of this, and that's the background. As far as the results, we saw that in the delta lines, which was basically where we already spliced the mRNA of XBP, so it was a positive control that would always be fluoresce green. We saw that to work in all of our models, and you can see that in figure 2, where it's the green islet. Versus in the non-delta lines, we saw that there was an increase in fluorescence, and most of the time of the transgenic fish that got heat shocked. However, it was very weak and it fluctuated in all of the different models. Yeah, figure 3 is a good example of it when it did work. As you can see, the increase in fluorescence there compared to the non-transgenic fish. But as you can see also in table 1, the expression rate was 25% within those that would show the positive heat shock response versus a 50% rate would be Mendelian. So there's clearly some form of silencing or something, some other mechanism going on that was causing this not to be Mendelian. Therefore, it wasn't reliable or a robust method slash construct. So that led us to know that we needed to keep on refining this model in order for it to be more reliable and before we can go and continue to test other genetic backgrounds and conditions. And so the first step that we'll be doing after this study that we completed will be to revise our model and possibly switch around the QF and QS system and possibly even swap it out for a single promoter cassette that may reduce the complications that we're seeing and possibly the silencing, which we may believe to be tied to the red heart screening factor, which has been seen to not always show up. And so maybe the red heart screening factor is causing some form of lethality that the zebrafish is trying to suppress and silence, which is interfering with our reporter system. And so, yeah, that's our findings in my project. Thank you for listening.